biological psych. In this part of the course, we're going to be having this kind of emphasis. And in biological psychology, we want to understand behavior. Well, we're thinking about behavior now in terms of what's going on in the nervous system. You know, why did somebody do what they did? Well, if they did not have an intact spine, they wouldn't have done that. If a neuron hadn't fired and sent a message to move a muscle, the arm would not have done that. If the brain hadn't computed the thought and the intention to do something, it wouldn't have happened. And so the interpretation is going to be basically biological and neurological. Now, to understand what's happening in the nervous system, we need to understand these 10 to the 10th neurons in the brain. And about 1 billion neurons in the spinal column. And then there are neurons coming out of the spinal column that send their axons to muscles. And there are specialized sensory receptors detecting touch, and temperature, things that are happening, sending messages, sending their axons to the spinal column, which is transmitting information up to the brain. And so let's diagram a sort of a standard neuron and discuss it here. So, your body is made up of cells. Human and animal cells have a cell membrane. We don't have cell walls the way bacteria do. Inside the cell is cytoplasm, a salty solution that's about as salty as seawater. So it contains, this is the cytoplasm. There is a nucleus of the cell containing the chromosomes that contain the genetic information that determines everything about you. Some of these chromosomes are turned on and some are turned off. This process, this area here is called the cell body. This long process here is called the axon. And this particular neuron is a motor neuron. The axon may branch out, and this one synapses onto a motor neuron. So this motor neuron synapses onto a muscle cell. This particular synapse, a synapse is when one cell comes close to another and communicates with it chemically. And so the neurons communicate with each other at their synapses. This particular synapse between this motor neuron, so this one is a motor neuron, and it's in the spinal column somewhere. And coming out of the spinal column comes the axon, and it attaches to a muscle somewhere. This particular synapse, where it's coming close to the muscle cell, is called the neuromuscular junction. And there are chemical transmitters in the synaptic cleft. It's a special type of synapse. And there are neurotransmitters that this cell is going to send chemicals that are going to influence the muscle cell and cause it to fire. So for you to lift something up, muscle has to become shorter. That's all muscles can do. They can relax or they can become shorter. If this muscle becomes shorter, this arm is a hinge here, so this muscle is attached to this bone and pulling this bone up. The muscle on this side relaxes. In order to go the other way, this muscle has to get shorter, this muscle relaxes, it goes the other way. There's sort of tree-like structure back here called the dendrites. So I'm not going to draw them all in, but down on this side we have dendrites. There are also mitochondria. Those are the sort of the ATP machines. These are the like cash machines. The cash of the human body is ATP. ATP is like money, you know, to the body. It's a kind of sugar that contains energy. The body needs the energy. The energy is processed in the mitochondria. The mitochondria are thought to have once been an independent organism that at some point in evolution was captured and taken over by animals, so that animals contain mitochondria that convert the sugar into ATP. Now if you stick an electrode, we have a galvanometer. We've got 2 electrodes here, and here's our galvanometer, and this arrow here is 0. Here's the negative side, so minus and plus. Here's on the positive side. There's no potential difference. There's no voltage between these 2 electrodes. They're both outside the cell. And we have a micro electrode that we can pierce into an axon. So we put one electrode inside the axon, and what happens is, what we can plot then, and when that happens, you see that there's a negative potential. Here's 0. This is negative, and this is positive. We have Benjamin Franklin to thank for the fact that electrons have a negative charge. So on your battery, you know, the little side with the bump on it on the standard battery, that's got a plus next to it. That's considered the positive charge end. The other end has a negative. If you connect a wire from one to the other, electrons flow from the negative end to the positive end, but according to Ben Franklin, the electricity flows from the positive to the negative, so people still say the current flows from positive to negative, even though the electrons are going the other direction. Well, he just had to guess one way. He went one way or the other. He had a 50-50 chance to get it right. He didn't know about electrons back in his day, but he's the guy that we credit for the one substance theory of electricity. So the voltage inside the cell, you can think of a cell here as like a little battery, where the negative end is inside the cell relative to the interstitial fluid, the fluid outside the cell. If you poison the cell, so here's 0 right here. If the cell dies, it goes up to 0 and stays there. So let's say we kill the cell, poison it. Basically, we kill the cell. Then the voltage goes back to 0. So the cell to be a living cell, you stick an electrode inside a living cell, it's negatively charged relative to the outside. This is called the resting potential. All right, let's now see what happens if we're not going to poison it anymore, so we'll come back here. We've got a living cell. A small stimulus produces a depolarization. It goes towards 0. So here we've got a stimulus. Now a bigger stimulus, or 2 small ones close together in time, produce a bigger depolarization. And an even bigger stimulus produces a depolarization that reaches a threshold. I'm going to show a dashed line here. If it reaches threshold, something new happens. We get what's known as a spike event. This spike event, the voltage inside the cell switches from negative to positive. So all of a sudden, this needle, this galvanometer, which had been reading negative, jumps over to the positive, and then it comes back to 0. What I'm plotting down along here is time, along this axis, and the voltage, or the electrical potential between the inside of the cell and the outside. So this is the voltage, I'm going to say, inside the cell relative to the outside. The inside of the cell is negatively charged relative to the outside. If you stimulate it, you get a spike event. Another word for the spike event is action potential. So we have the resting potential. It's negative. The action potential is positive. Another word for the action potential is a spike. And people also use the word, they say the cell fires. We're going to be talking about spike events and cell firing. Right after the cell is fired, if we give it another stimulus, it's harder during this period of time to get it to fire again. It's known as a refractory period. And during this period of time of about 1/1000th of a second, when it goes actually below, before it comes back up all the way, it's in what's known as an absolute refractory period, which means the cell can't fire again immediately after it's fired. So this is the absolute refractory period. And that's about 1/1000th of a second. And that means that the maximum firing rate for a cell, how many times can it fire per second, is about 1,000 times per second. So it's hard to get, if not impossible, to get a cell to fire again. Now the way to think about the spike event, the height of the spike is independent. Once it reaches threshold, the height of the spike is independent of the magnitude of the stimulus. You may get a lot of spikes from a big stimulus, but you don't get big spikes and little spikes. And this is known as the all or none law. All or none law says that when the cell fires, it fires. If it doesn't fire, it doesn't fire. You could think of this as, and that's why they use the word firing as a metaphor. Think about firing a gun here. We don't want to be associated with the NRA here, but some guy from the NRA has got his gun and shooting at a target. Hopefully just a target, not a policeman or somebody else. Squeeze the trigger a little bit, what happens? Well, you get a depolarization of the trigger, but it doesn't fire, right? So you squeeze the trigger and let it go and it goes back. You squeeze the trigger a little bit harder, it goes and it comes back. But if you reach a certain point, what happens? The gun fires, right? And the size of the bullet that comes out is independent of how hard you squeeze the trigger. Either it goes or it doesn't go. So if you squeeze it too little, it doesn't go. But if you squeeze it hard enough, it goes. If you squeeze it even harder, it doesn't go any bigger. So the muzzle velocity and the mass of the bullet doesn't change depending on how hard you squeeze the trigger. So that's the idea of the all or none law. It either fires or it doesn't. The magnitude of the spike is independent of the stimulus, given that it fires. So once you know that the cell is fired, it's going to fire. Now, how do you think about this firing, the spike event? Well, the spike event is usually initiated here at what's called the axon hillock, and it travels in one direction only. In a normal cell, it travels from where the spike starts down the axon in one direction. Some communication devices communicate in both directions. So if I had a string radio, has anybody made a string telephone, or two cans and a string? Anybody ever made one of those? Yeah, okay, this guy's had one. Here's a can with the string on the end. You get two tin cans, you poke a hole in one end, poke a hole in the other end, you attach a string to a nail or something, you pull it through the hole. I give him one can here. Then I stretch it out. Got a string here, we got it stretched. Be even better if the string is wet. It's attached to another nail on this side, and then I can talk and he can listen. Listen, you can hear what I'm saying. He says yes. Go ahead and say yes in there. I don't hear very well.
Yes.
Okay, I heard that. All right, so the information can go both directions down the string, right? We can communicate in two directions on the same string. The nervous system does not work that way. Don't think that it does, because that idea people come with, they bring that idea in, that it's like a string radio. Not a string radio. Every neuron is carrying information only one direction. Either it's carrying information, say, outside the central nervous system, outside the brain and the spinal column, carrying information from the brain, from the spinal column to the muscles, or carrying sensory information, that's in the motor system meaning what makes you move, or the sensory system, meaning it's carrying information back from the muscles or from the skin to the brain. Now the spike events travel down here at a finite speed. What is the speed of transmission? Well, we can set up two of these. Start a spike event and look at when it goes off here, and then when it goes off here, and when it goes off down there. So we can time how long it takes. And the speed of transmission differs for different axons, for different neurons in the body. It can vary from as slow as 1 to 2 meters per second in an unmyelinated axon to as high as 800 meters per second in a myelinated axon. What do I mean by myelinated? I mean some axons are wrapped with fat cells. And your brain is loaded with fat cells, wrapping axons. Somebody calls you a fathead, don't be insulted. It's literally true. Your head is full of fat. And you need that fat. Your head is full of cholesterol, high cholesterol fat. You need it. Okay? Don't listen to people that say you should be on a zero cholesterol diet. You shouldn't have any cholesterol in your body. Well, the problem is, people eat too much fat. Then the theory is they get too much cholesterol in the blood. Maybe they inherit a tendency to have too much cholesterol in the bloodstream. That's bad for your heart to have too much cholesterol. So too high a diet, fat diet, not healthy for you. But for children, they need about 1/3 of their calories to be from fat because they got to build a brain. And in their brain, there's lots of fat cells. The diseases of these fat cells create severe neurological disorders. So you definitely want to have healthy fat in your brain. Now the fat is called myelin. I'm drawing it this way, but a way to think about it is it's wrapping itself around. So there are cells that wrap themselves. There's a cell that's wrapped itself around here, and another fat cell wrapped itself around here, and another cell that wrapped itself around here. And the theory is that the spike event jumps the myelin, and that causes it to be a faster transmission velocity. So the myelinated fibers carry the information faster than the unmyelinated fibers. The unmyelinated fibers in a human are associated with pain. Some organisms only have unmyelinated fibers, like squid. They have to have a big, thick axon in order to have a quick reaction to something. And the big axon in a squid, what does a squid do that it needs to do fast? When it squirts out ink, it swims away. So the big axon that runs down the squid's body is a thick axon to be faster so that it can make that reaction quickly. Humans, though, and higher animals evolved this trick with the fat, which means that the speed of transmission is much faster. Now, touch is myelinated fibers. Contact with the skin you can detect with, but pain is being transmitted by unmyelinated fibers. So this may explain some things to you. In the case of a person, you can have a person who's 2 meters tall. That's about 2 meters right there. You're about 2 meters, right? Maybe a little more, I don't know, yeah, somewhere close to 2 meters. So a 2 meter person, and here's a guy who's probably done this before. I don't know, maybe not. You stub your toe, and you know you've stubbed your toe before you feel the pain, right? You stub your toe at 800 meters per second, that's pretty fast. He knows right away, oh, I've stubbed my toe, and he has time to think about it and know, here comes the pain, right? So he would testify to that. Some of you have also noticed this discrepancy. You realize something has happened before you feel the pain from it, because the pain is coming information. Now, another way to think about that, of course, is Everything you perceive about the world is in the past. It's already happened. People in astronomy are familiar with this already, right? Something happens in the Andromeda galaxy today, we won't know about it for 200 million years because it takes light that long to get to us. But even after something impinges on your nervous system, it takes time for it to get from being received in the eye to going into the brain for you to realize what's happened. So everything you perceive is always in the past. And here there's a discrepancy between two events that happen at the same time, stubbing your toe and that happening. All right. So let's review a few of the properties. Before I erase this, let me make sure there aren't questions about this. So here's your standard neuron. You should know about the cell body, about the nucleus, which contains the DNA is in there, right? Your chromosomes are in there. They're called chromosomes because they're the colored bodies that were theorized by Mendel to be there and finally seen in 1905 when they put some dyes in there so they could see the chromosomes, and they realized you got one set from each parent, right? So your DNA is in the cell nucleus. You've got the cytoplasm, that is a salt solution that's about as salty as seawater. We have a negative charge inside relative to outside. That's the resting potential. We also have the action potentials. We can have spike events, can have up to 1,000 spikes per second in a neuron because there's an absolute refractory period. There's also a relative refractory period during which it's harder for the spike event to happen. It takes a bigger stimulus to get it to go again. So there's a relative refractory period, but because of the absolute refractory period, you just can't get cells to fire all that fast. What's creating that potential, the theory is that there's a sodium pump pumping sodium ions. When you take salt is sodium chloride. Sodium chloride is NaCl, and when you dissolve it in water, the sodium and the chlorine become ionized. So that means that the sodium is carrying a positive charge and the chlorine is carrying a negative charge, and they dissolve in the water. And the body, because it's alive, is pumping out sodium. And this sodium pump, because there are large negative ions that are trapped inside that are not going, we have semi-permeable membrane. This cell membrane is called semi-permeable. Which means some things can go through it and some can't go through it. And sodium is being pumped through it, and then somehow it's changing. The membrane is actually changing and letting the sodium back in, which is allowing that voltage to change direction and actually become positive. An example of a semi-permeable membrane, anybody taking chemistry that's in here and work with the Nernst equation? Nernst equation is an equation that says if you have a semi-permeable membrane and you have salt water on one side and regular water on the other side, or you have salt water at two different concentrations, there will be a voltage difference between the two. And from the Nernst equation, you can calculate what that voltage difference is going to be. And so you can put different chemicals on two sides of something that's semi-permeable. Some of you may be familiar with a semi-permeable membrane that's inside your gut. So your intestines are semi-permeable membranes, right? What's going on there? You eat food, it goes through your body, goes out of your body, right? Never goes into your body except through the semi-permeable membrane into the bloodstream. So you're able to digest food by putting chemicals in there that react with the food, break it down, right? The food nutrients go through the semi-permeable membrane of your gut into the bloodstream and thereby give you nutrition. And what are you doing with that nutrition? You are burning a fire that's keeping you warm, right? You've got an oxygen fire. You're breathing in oxygen, burning up sugars, these ATPs we were talking about, breathing out carbon dioxide. So you're creating this carbon fire is going and you're warm. Your body is running at a certain controlled burn. It's not quite like setting a piece of paper on fire, which would be 450 degrees Fahrenheit, but you've got a controlled burn. It's being burnt under with special enzymes that allow that to happen that way. So this semi-permeable membrane inside your gut, other people have used them for condoms. So you can take a sheep and take its gut out, tie, cut it into tubes and tie off one end and use it as a condom, and sperm won't go through it, so it can prevent pregnancy. And the syphilis germ can't get through it, so it'll prevent you from catching syphilis. But unfortunately, HIV can go through. HIV is a small enough virus, it can go through the semi-permeable membrane. So the sheep intestine condoms don't prevent HIV. That's why they recommend latex condoms for prevention of spread of hepatitis and HIV, sexually transmitted diseases that are spread through sex. But it will prevent pregnancy and it will prevent the spread of gonorrhea and syphilis. So some diseases that sheep's intestine will prevent. So we've got a semi-permeable membrane, and the Nernst equation you can use to calculate what the voltage is, and people have actually done that work in this field of neuroscience. It might be called physiological psychology or biological psychology. You're going to hear all those words being used: neuroscientist, physiological psychologist, and biological psychologist. All right, any questions about the cell and membrane potential, the sodium pump? Let me talk about a little bit more about how cells interact with each other. Any questions here before we go on? All right. Now, cells interact with each other at the synapses. So here's cell A, and it's sending its axon over here, which might branch off, and here's its terminal button. And here's the dendrite of cell B. There's lots of dendrites. So here's cell A, we call this. And here's cell B. Here's its axon. We're going to stimulate cell A, and we're going to look at what happens to the firing rate in cell B. This is in living tissue, so it's not going to work if we have cells that are dead. This is going to work if we have living tissue. So we're going to record from B. So we've got our one electrode is inside the cell and one electrode is outside the cell, and we're looking for the spike events. First thing you notice is we leave A alone, and you've noticed there's a firing rate in B. So there's what's known as a spontaneous firing rate. So all cells, so no stimulation. In cell B, we've got a certain firing rate. I'm making these marks to indicate the spike events. Okay, so each one of these little lines is one of those spike events that I was showing you before. You may attach, and you're going to see in some of the videos that I'm going to assign to you to look at, they're going to attach the device to a speaker so that they can hear pop, 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 pop coming out of the speaker and hear the spike events happening in the cell. And that way the neuroscientists can be working with their preparation and they can be listening. They don't have to look at an oscilloscope to see the spike events. In the old days, they used to have it paper being pushed through by a motor and a pen making these on paper. So the spikes were originally, you were looking at the voltage was being read on paper and the paper was being pushed through. These days, it's on an oscilloscope screen and the data are being stored into a computer, and so they can recreate the graph. It's being stored inside a computer. So you just we stuck our electrode in here to this cell, and we're hearing from B a certain firing rate. And now we stimulate A, and the firing rate might go up. Up. All right, so it may increase. If stimulating A, which causes A to fire, increases the firing rate in B, we say that this is an excitatory synapse. So this synapse is said to be excitatory. Now it could be that we've got some cell C here. So let me branch off. So maybe cell A comes to another cell, cell C. And we're going to now record from cell C. We stimulate A. And we might see, and here's the spontaneous rate here. Let's say no stimulus here. And here's the firing rate in C. So here we're recording from B. And B increases. Here's the spontaneous firing rate in C. And now we stimulate A. And we see that C shuts up. Up, was going, up. And we turned on A, up. Could decrease. So in this case, C decreases. And we say, in this case, that this synapse is inhibitory. So stimulating A could increase the firing rate in another cell, or it could decrease the firing rate in another cell, or it could do both. In this case, A is exciting B and it's inhibiting C. And this is going to be one of the reasons that even though the images in the back of your eye are blurry, you see sharp vision. And it has to do with the lateral inhibition of the neurons inside the eye. So you're able to detect, if we put a very vague stimulus on your arm of vibration, people can detect the point of maximal vibration, even though it's a very vague stimulus on there. People are able to see sharp corners on things, even though the visual image inside the eye is blurry. And that has to do with what's known as lateral inhibition. The fact that you have some cells exciting and some cells inhibiting. And the brain works with both excitation and inhibition. Let me review, we're going to review a few of the basic terms, and we'll be continuing with it next time. Let's make sure I said a few things about each of these. We can get this. While that warms up, I'll tell you about this picture right here from the book. So here's what's going on at the neuromuscular junction, or at the synapse. So they're showing some mitochondria in there, and there are these vesicles which are packets of neurotransmitters. When the spike event comes down the axon, so here's the one neuron, here's another neuron, here's the axon, and here are the synapses down here. Into the synaptic cleft, that's the gap between the two cells. So the two cells one doesn't really go inside the other, it just comes near. And it releases chemicals. That's the way the cells talk to each other chemically, by releasing chemicals into the synapse. It's releasing neurotransmitters that may excite this cell and make it fire more, or it may release another neuron may release chemicals that inhibit this cell and make it less likely to fire. So the neurotransmitters could be excitatory or inhibitory. They could increase the firing rate or decrease the firing rate. Certain drugs influence what goes on in the synapse. One of the drugs that influences what goes on in synapse is curare. Another drug that influences what happens at the synapse is strychnine. Both of them can be used as poisons. Curare is the poison that the South American Indians use for hunting in the blow darts. So I will simply tell you, if we'll finish up with the blow dart, we'll continue next time. The Indian chemist of the tribe mixes up the curare. It's made from a plant. You can taste it. As long as you don't have an ulcer or an injury in your mouth, you can eat it. It doesn't kill you that way, but if it gets in your bloodstream, it's poisonous. You dip the arrows in it, put it into the blowgun, find a tasty monkey, aim high. If you aim right at it, gravity will pull your dart low. It's like students aiming for a certain grade. Students have a tendency to fall. Aim at the monkey high. You shoot the monkey with the curare. Pierces the monkey's body, gets into the bloodstream, the monkey goes limp, falls out of the tree, stops breathing, and you can have him for dinner. All right? I'll tell you more about that monkey next time. It's an important story for understanding neurology.